So I'm here, but I might want to attend online and in the lab. Are you okay?
Yeah.
Okay.
Should be.
And then probably there will be some people from here as well. But yeah.
Well, Martin will come here. Good afternoon. Great to see you.
Good morning.
Well,
How are you doing?
I'm doing fine. I'm Father Spenter. I'm the one that sent you the links and things. And I see everyone is also online already. So
there we go.
Kelsey is in the waiting room, so we'll get her in a moment. So, and then we can allow you to share and to see if it's possible.
All right. Sounds good.
I've
Good morning, Kelsey.
Hi, Mike. How are you?
Pretty well. It's just now dawn here, so there'll be more light when my sun comes up. So, but sorry for the black backlight. I don't have my Zoom stuff anymore.
No problem.
So should I go ahead and share screen or just to try it out?
Yes, please. I think, Fannis, are you there or have you frozen?
Oh, he's there. I could see him. All
Okay.
right.
Oh, there we go. Okay.
So I'll try it this way if that works. Let's see. Do you guys see that?
Yep. Looks good.
All right, I'll just do the desktop share. That's a little bit, I find that's a little bit more foolproof than trying to capture the actual window when making, they used to work and then now like it doesn't capture the presentation window or something half the time, I feel like. So we'll just leave it. We'll just do this. All right, well, I'll stop sharing so that, let's see.
I've had too many bad experiences if you have a second screen that's attached.
Exactly.
And so that I have disconnected the second screen, especially because we've got load shedding in a few minutes and then it's going to disconnect halfway through and it's just going to be a mess.
Oh gosh.
All
I
right.
thought you guys would be spared from load shedding being in the Cape.
Not entirely. We're spared from the from the lower stages. But luckily it's fine.
Okay.
So let's have a look. Share desktop. Cool. I'm assuming you can see my screen.
Yeah, looks good.
Yeah, it looks good over here.
Awesome.
Hello, can anyone hear me? I don't think.
Hi,
Yep,
Henny.
hear you.
Hello.
Can you grab my phone? Yeah, thanks.
Say hello. Hi. I cannot
there.
hear anyone. I don't know if I am having a setting that's a problem. I just see everyone, but I cannot hear.
Okay. Well,
Let's see.
Want to finish up your stack, and then you're supposed to. Oh, you've got 5 minutes.
Okay. Yeah.
so Henny, do you want to try sharing your screen?
Don't prepare for that.
If he can hear you.
All right. Oh, what's happened?
Then you'll have shoes.
Okay.
Okay. Okay.
Okay, here, you got it.
Share screen. Okay.
Mhm.
Hey, Brucey, you're supposed to pitch back and get on socks, eh? Coming pretty soon.
Cool.
Okay, more settings.
Mm.
I'm gonna real quick try to just share the actual screen, the actual presentation, because I feel like if I do the desktop share, you see the Zoom bar at the top, right? So
Yeah. Okay, cool.
let's see if that works better. Oh, does that look good?
Yeah, that looks
All right.
that looks good.
I'm just gonna
Cool.
pause that and then I'll restart it when things get going.
Sounds good. Hmm.
Or stop it. There we go.
Hi Kelsey, I've just made you the co-host as well, so that if something happens here on our side. Things can go continue.
Cool. Thanks, Venice.
Okay, I can hear. I think I can hear
Hey,
everyone.
that's great.
Yes, well, we
Okay,
can hear
fantastic.
you.
Hooray!
Okay, winning.
Sorry about
Yeah.
that. Okay.
It diffuse, yeah, it doesn't diffuse. And let us see if it look okay like this.
One, you must just mute. Juan, can you mute?
Actually, it look not bad. Actually, yeah, no, actually look not bad.
Okay. Venice, did you kind of run through the general progression or not yet?
Sorry?
Did you run through like okay, here's how this will work?
No, we haven't done that, but
Okay.
basically, so I'll just at 4 or shortly after 4, depending on how people come in, just welcome everybody, and then I will hand over to you. You can then introduce the speaker, and then we will deal with the talk and then do the questions and introduce the other two. So I'll do the first bit and then it's over to you.
Sounds good. Cool. Thanks.
Right, Kelsey? So Martin is also here.
Hi, Martin. I was debating going to Wits for this, but our Wi-Fi was not very good today.
Hey, that's a different Martin.
Oh,
Yeah, sorry.
Yeah.
Mine.
oh, sorry, I thought it was Martin.
I'm sitting next to you from this.
No, I thought you were also logged
Yeah.
in.
No, I'm not.
So we've already got one other attendant. Thank you, Martin.
Thanks.
Martin. Okay, now we just wait for people to attend. So as people come in, I will allow them in. So we ready. But numbers are still good. No. It's Dharma. Hi, Vitama. Thanks for joining. Kelsey, did you attend the workshop or not? No. Vitama, any feedback just like here from the workshop? It sounded like it went well.
Thank you, Fanas, and good afternoon. It was fantastic. Fantastic is the word. Really good.
That's great.
36 attendees drawn from at least 10 institutions, lots of good discussions, nice setting, nice food, nice drink. Thank you. Oh, very good.
That's great, and then at least we could recover those funds.
Yes, and Ruan was one of the attendees. Maybe he has an opinion on it.
It was a lot of fun. No, it was the venue was really nice. If a bit windy, but no, it was great. Like we all learned a lot. Like it was a very busy few days, but it was thoroughly enjoyable, and it was very interactive. So we worked a lot with people brought problems that they had to discuss with Sandy and Nick, and that was really interesting.
Well, that's great to hear. So we'll just wait a little bit. We still have a minute, and as you all know with Zoom meetings, everybody waits until the last minute or thereafter.
5 minutes cost.
So we'll start a little bit later. Just to get everybody in. I don't know if you've seen on what used to be Twitter, X, but our young people have been very active in tweeting or Xing this talk. So that was quite nice.
Oh, that's great.
So I should encourage everybody to follow our group on Twitter. On X. Give a minute or two, stop. I think let's get started. The one thing I know Ruan has indicated that he doesn't mind the recording being shared. Mike, do you have any objections that we share this on YouTube? So you're fine with that?
I'm fine with that. No problems.
Thank you very much. And Henny, you?
Also, no problems at all. I'm happy with that.
Thank you. Then we will share this on the YouTube channel as well. So I think let's get started. Welcome to everybody. We've basically already had a chat here amongst everybody that's here. Thank you very much for joining us today, and a special word of thanks to Mike. Getting up early in the morning to give us a talk. We really appreciate it. And then to our two local guys, Ruan and Eni, thank you very much also for being willing. I think it is for a number of people quite an interesting topic, polyploidy, and therefore we're quite excited to listen to you. In terms of the society, we've just had a chat and everybody said that that workshop was great fun, and that's what a workshop should be, I think. You know, if nothing else, it should be fun. But at least, as you said, the part of the fun was the fact that you could deal with problems and learn from there. So we're very happy that that also went very well, and thanks to Matamu and the group that organized that. And then with that, I'm going to give over to Kelsey.
Thanks, Dennis. Hi, everyone. Thanks for tuning in for our last SASIB webinar of 2023. It's my pleasure to introduce Mike Barker, joining us from the Western US. Mike is an associate professor at the University of Arizona and is well known for his work on polyploidy's role in contributing to the ecology, evolution, and diversity of life. So his lab integrates both computational and evolutionary genomic tools with phylogenetics to understand the origin of biodiversity. And notably, Mike is the progenitor of the Polyploidy webinar series that kept me and many others engaged and enthusiastic during lockdown. So thanks so much for doing that, Mike. I don't know if you quite understand what a key part that played in keeping me going. So thank you for starting your day with polyploidy as we end ours with polyploidy, and I'll hand it over to you. Thanks, Mike.
All right. Well, thank you, Kelsey. It's wonderful to be here. I'm gonna go ahead and share my screen, and hopefully this works this time again. All right. There we go. Everybody, hopefully you can see that. Yeah, this is really an honor to be invited to speak to you, everyone, today. And it's been we've all had I think a lot of experience giving seminars and talks on Zoom now, and so this is a great way to do it. Although it would be nice to be in South Africa. Hopefully, sometime here we'll get over there and visit and get to see the really wonderful diversity of life there. And today I'm going to talk a little bit about our work on polyploidy and the evolution of plant diversity, as you might imagine here from my title slide. And I'm going to really focus on some of the recent work that we've been doing. Part of this published, some of this soon on bioRxiv, hopefully in the next week or so. But really focusing on understanding some of these large-scale macroevolutionary dynamics and how we connect some of these patterns of polyploidy across the tree of life to as well to contemporary plant evolution. And so I'm going to go ahead and get started here. So for those of you that aren't familiar with polyploidy is speciation by genome duplication. It's been recognized for over a century now as a mechanism of speciation and evolution in plants in particular. One of the classic sort of networks of polyploid speciation is that of the brassica, what's known as the triangle of Yu or the triangle of Wu, worked out by Wu Jiangchun back in the early 20th century. And this is the kind of thing we often see right in clades of plants. You get diploid species here in these corners, in this case Brassica nigra, Oleracea, and Rapa, that all make hybrids with each other and that also undergo genome duplication at some point in that process to become allotetraploids. And so here we see, for example, on the bottom of this triangle, Brassica oleracea and Brassica rapa, and their allotetraploid Brassica napus. And this is a pretty common mechanism of speciation in flowering plants in particular. And there are many economically important polyploid and hybrid taxa out there on the landscape. Everything from the cotton that is potentially in your jeans and your clothes to peppermint and spearmint and blueberries and oats and potatoes and lots of other things, including ancient polyploidy in humans, but also even polyploidy in fishes. Fishes are in some ways the plants of the animal world with regards to polyploidy. And this in particular is a big triploid heterotic trout. I think this is the state record for California, which is heterosis will get you there, I guess. All right. But outside of these economically important plants and animals, polyploidy is really common on the landscape. So in our surveys over the years, going back to our PNAS paper in 2009 to papers in 2011 and 2016, we've had pretty similar frequencies of polyploidy across flowering plants. In particular, if you look out, just go out of the landscape and just ask how many species have double the number of chromosomes relative to their diploid, relative to their base, you see about 1/3 of plant species are consistent with being recent polyploids. And when you do a little bit of math and ask, what percentage of speciation events appear to be accompanied by polyploidy in these phylogenies, it's about 31% for ferns and 15% for angiosperms. So a lot of plants have polyploid genetics on the landscape, 1 out of 3, roughly. And a lot of plant speciation events, a lot of diversity itself derives from this process of whole genome duplication. And if we sort of drill down further into polyploids, and we ask, polyploid species come in 2 different varieties. There are allopolyploids and autopolyploids. Broadly speaking, it's really a continuum, but you can sort of think about binning them in those 2 ends of the spectrum of polyploidy. Allopolyploids come from hybridization and genome duplication between 2 divergent lineages or species, and they make up about half of those identified polyploid species in the landscape. But nearly all of them are named, something like 95%. But the inverse is true for autopolyploids. Autopolyploids are species that arise, or are polyploids that arise from self-duplication. So duplication within a lineage, meiotic errors, for example, that might go on to propagate a underduced gamete that goes on to propagate a new lineage of tetraploids or hexaploids or whatever the ploidy level is within that lineage. But many of these are not described. I believe something like only 8%. But they are a substantial fraction of polyploid taxa on the landscape if you think of them as categories. And so polyploidy appears to come in these sort of two flavors, and in our estimates, pretty nearly equal frequency, although it varies quite dramatically across genera, from some genera having almost all allos to some having almost all autos, with a nice even distribution. And that's a whole research career right there, just sorting that out. In addition to these recent polyploid species, plants have a deep history of many rounds of ancient whole genome duplication. I love this plot. This is from the Vitus paper from back in 2007. But it's a nice sort of cartoon, I think, that kind of exemplifies these rounds of multiplication and rearrangement and fractionation that happens. For example, here in Vitis vinifera's genome, you can see it's like these focus on these orange chromosomes here, 6, 8, and 13. And you can see there are these clear sort of 3 duplicated chromosomes, these 3 homologous chromosomes from the eudicot hexaploidy that in Populus and Arabidopsis have been duplicated again and again, and then fractionated again and again and rearranged, so that even though Arabidopsis has only 5 chromosomes, it's gone through a couple more rounds of polyploidy compared to Vitis, but then lots of rearrangement and lots of loss of chromosomes to get down to its contemporary genome, which means that plant genomes are just fun, right? There's a lot of rearrangement and duplication happening. But also that there's a great legacy of paleopolyplane plant genomes when you look at the genes themselves. So if we look at the fractions of genes in plant genomes that are derived from these ancient polyploid events, it varies dramatically from more than 2/3 in soybean to as few as 25% or so in Arabidopsis. And these are what we call in our lab at least paleologs, these genes derived from paleopolyploidy. And these are genes that are duplicated in those events. And so polyploidy has created left a significant legacy then in not only the structural organization of plant genomes, but also in the diversity of gene families themselves. And for these reasons, and for many other hypothesized reasons, polyploidy has been proposed to be a significant sort of source of diversity, and maybe an important driver of the evolution of plant diversity across the tree of life. And this is a really fascinating question we're going to get into a little bit more today from some unique angles. And one of the things that we're going to talk about today is how we reconcile our observations that there are multiple rounds of polyploidy in the history of plant evolution, but with our estimates that many polyploid species actually have lower net diversification rates than their diploid relatives. And try to sort of put together all of our perspectives on polyploidy and diversification and the distribution of ancient polyploidy across the tree of life. And then at the end there, ask a little bit about what are the impacts that these ancient whole genome duplications have had on diversity and adaptation of contemporary plants. And so trying to draw a little bit of a line from those macroevolutionary events that happened millions of years ago to contemporary evolution out there in the landscape. So just starting with this first question, if we, as I just mentioned, plants have this amazing history of ancient polyploidy, and that there are many, many rounds of whole genome duplication across the history of the flowering plant phylogeny. And we uncovered a lot of this in a large survey of green plants that came out a few years ago with the 1KP project. Some of you may know there's a really big group of us that worked on this. And in this survey, we analyzed a bit more than 1,000 plant transcriptomes. It was something like 1,400 different species, every family of flowering plants represented. And we used those data in our lab to ask when, where, and how many whole genome duplications have happened across the tree of life. And this was a team effort at the time, led by a combination of undergraduates, visitors, grad students, and postdocs, all led by a PhD student in the lab, Zhang Li, and really 3 undergraduate students that were critical to the success of this project: Sally Galuska, Chris Reardon, and Thomas Kidder. And see Thomas here with this giant scroll. This is our, even though we do everything on the computer, we still printed this giant phylogeny out, and you can see it's rolled up down there. I have it actually over here on my desk, with this giant printout of the 1 kp phylogeny to do manual checks. So this is our scroll of polyploidy, as we like to joke. And in that big analysis across all of green plants, we found, and we added some other data, so we had more than 1,500 green plants to this data set. We'd found about 245 ancient whole genome duplications across that phylogeny. And if you look at the branches in the flowering plant phylogeny, for example, we see that about 13% of those branches have an inferred whole genome duplication on them. I think the punchline from this is just that this is the sort of high elevation view of this, is that the punchline is there's lots of polyploidy. And we can quantify that in different ways. I think one of the, for me at least, and as you'll see for doing the sort of these later analyses, one of the important ways to think about this is just how many rounds of whole genome duplication have happened in each lineage of flowering plants, or in each species of flowering plants. So what we do here is we just look at the overall tree and go back from tip to root and count the number of rounds of whole genome duplication in these lineages. And what we see for flowering plants in general is that they have around 3 to 4 rounds of whole genome duplication. And that's comparable to ferns, but many other lineages of eukaryotes, or excuse me, of green plants have many fewer. But just focusing here on the flowering plants, we see that there are 3 to 4 rounds of polyploidy on average in your average flowering plant. But we also know that recently formed polyploid plants appear to diversify have lower net diversification rates than their diploid relatives. This is from work that we published a while ago now, back in Science, and then revisited this in an opinion paper as well. And what we see here, I think perhaps even easier to see in the box plots on the right, is that if we look at across these roughly 50 genera that we analyze net diversification rates in, we see that polyploid species on average have lower net diversification rates than their diploid relatives. So how do we explain this, right? If polyploid diversification is generally low, then how can there be so many ancient whole genome duplications throughout the tree of life in flowering plants? And that's what we're going to work on today. So there are many possible explanations for this disagreement between these two results, or this apparent disagreement. The first is that, maybe our estimates are wrong. And this is certainly something that we've all discussed in the literature over the last decade, is that the diversification rate estimates could be wrong. I'm not going to get into that in any detail here. You can feel free to read our exchange with the Duggingham, Soltis, and folks and new phytologists. Suffice to say, I don't think that our diversification rate estimates are wildly wrong or off, as we'll see later as well. But it certainly could be the case that they don't reflect the whole reality of what's happening. We also maybe are on the ancient polyploid side of things. Perhaps we've inferred too many ancient whole genome duplications. This is certainly something that some people have argued, but other people have argued that we found too few. I feel like we found whatever our tools have said there are, personally, in that I don't think that it's wildly wrong. And we've actually, I don't have it in here, we have a completely new analysis of ancient polyploidy across the whole green plant tree of life, and it's nearly identical to the results we have today. And I'm happy to pull those slides up, or those figures up, from my lab Slack, which we just got yesterday, at the end, if anybody wants to see those. Okay, so I don't think the estimates are entirely wrong, but they could be. All right. There are other reasons, though, biologically, that we might see this disparity. It may be that polyploids have had higher diversification rates in the past, and so that contemporary polyploids diversification rates compared to diploids is low. And there is some argument that there may be a wave of polyploid events near the KPG boundary. But regardless of whether there's a concentration of whole genome duplications at any particular point in time, it's likely that diversification rates have varied over time and in space as well across the landscape. And so we may see that plays a role here in this apparent disagreement between these two results. It's also true that polyploid diversification rates could vary with other traits. And there are 2 nice studies that explore this. One from Rosanna Neil Ferguson and colleagues, looking at the interaction between or among polyploidy breeding system and lineage diversification in the Solanaceae, and one from our group led by Christian Roman Palacios, looking at the Brassicaceae. And just to sort of highlight how this works, I'll just give a little bit of overview of Christian's work. In this case, Christian collected nearly over 50% of contemporary species in the Brassicaceae into a phylogeny with chromosome counts inferred, and then inferred, with chromosome counts, then inferred ploidy level using a couple of different approaches, using chromovol as well as what we call the Stebbins fraction from those data. And using that, he was able to infer diversification rates and its interaction with polyploidy. And one of the things we saw is the same kind of thing we see in our 2011 science paper and in other places, and same thing that Rosanna and other folks have seen in Solanaceae, which is that diploids generally have here, we see the same thing, a slightly higher net diversification rate than the polyploid relatives. However, if you look for complexity in this data, in this case using a tool called HISI, which looks for hidden states that may be explaining some underlying structure in the data without you knowing what those are a priori, right? It's a very powerful approach to identify these kinds of associations. When we look at this, we see that there's actually a really strong state. We're not sure what it is, mating system or annuality, perenniality. It's not entirely clear what the state is itself. But if you are a hidden state B and you are a diploid, you do really well compared to the polyploids over here in hidden state B. And if you're hidden state A, if you have that trait, whatever that might be, if you have the state A and you're a polyploid, you do slightly better than those diploids. And so a lot of this pattern is potentially being driven by this underlying distribution of another trait that drives this. So the punchline is that the interaction between polyploidy and traits and diversification is complex, and so we don't expect there to just necessarily be a straightforward answer. But on average, we do see that polyploid species have, across the whole tree, at that high level perspective among contemporary species at least, that polyploid species have lower net diversification rates than the diploid relatives, but not always. Depends on the combination of traits. But the final sort of take here is that there may not be any conflict and that these estimates actually largely agree. And that's what we're going to explore today. And what we're going to do is use simulations to test, if this inferred number of whole genome duplications that we observe across the whole flowering plant phylogeny is consistent with estimated net diversification rates from contemporary inferences. Again, these contemporary inferences of estimated net diversification rates are pretty similar across different studies. And we also see similar numbers of inferred whole genome duplications across the phylogeny from different studies. And so I think there's merit here to trying to reconcile these and see how we can make sense of the results. So to do this, we're using a simulation framework based on BISI. So this is the binary state speciation and extinction model framework. We did this in Diversitree. And in this model, this is work led by Xing Zhan and a graduate student in the lab, Choyu Zhang. And in this model, what we're doing is allowing there to be diploid and polyploid species that both have their own speciation rates and extinction rates, as we see here, and that diploids can polyploidize. There's a rate of polyploidization. They can polyploidize to become polyploids. And likewise, polyploid species can go through diploidization to become diploids again. And so we run these simulations through this model and count the whole genome duplications in the history of each tip, just as we've done before empirically when we look at the number of polyploidies across the phylogeny. And so you get something that looks like this in these trees. You can have diploid, diploid, polyploid, and we count back the number of transitions that have occurred at each of these lineages, just as an example of what these simulated trees look like. We can we actually have a lot of data from our empirical analyses that we could use as parameters for these simulations. So, for example, for our this sort of first, and I'll get to that in a second, I guess. But for our first example here, I'm just going to explore what this parameter space looks like and how these different parameters impact the model itself, or the distribution of polyploidies across these different parameters. And for this first set of exploration simulations, we use a range of arbitrary net diversification rates, everything where polyploids have much higher rates of diversification compared to the diploids, as well as situations where the polyploids have much, much lower diversification rates compared to the diploid relatives. In this set of exploration sims, we can, for example, set the polyploidization rate to 0.15, which is our empirically estimated rate of polyploid speciation from Wood et al. 2009. We don't have an estimate, though, for diploidization rate. We don't have an empirical estimate for what that is. It's always sometimes a little bit harder to get these kinds of extinctions or rates or these other things on these that sort of downward side of the curve. And so the diploidization rate itself here is set across an arbitrary range of values from relatively slow at 0.2 up to 0.8, a relatively fast rate of diploidization. In this set of exploration sims, we just run 1,000 taxa with 1,000 replications for each combination. And so this is just to give us some perspective on how these different parameters interact and drive changes in the model. And we get a panel of things that looks like this. So and I'll go through a box here in a second, but basically on the y-axis, we have the number of whole genome duplications. These are the median across simulated lineages. So this is, again, counting from that like tip to root, the number of polyploidies that have accumulated within each species in these simulated phylogenies. And generally speaking, when we go from the bottom left here is the case where polyploids have much higher net diversification rates than the diploids. To the top right, the case where diploids have much higher net diversification rates than the polyploids. And if we look at a single box, what we see here is on the y-axis, or we have that number of whole genome duplications that are inferred. So you can see that in the box. So for example, this set of parameters strongly in this heat map, there's 2 rounds of polyploidy on average in these species, up to 3, for example, in this part of the parameter space. On that x-axis, then we have this rediploidization rate. So this is that rate of diploidization of the polyploid species going back to being diploid from slow to fast. All right. So what do we see? Well, generally speaking, if we look at this set of exploration simulations, we see that there are more ancient whole genome duplication when we have higher rates of diploidization. You'll see that in every single one of these boxes, there's generally a trend from the bottom left with that line of color indicating more rounds of polyploidy going to the right. And so when rates of diploidization are higher, that's when we see more rounds of polyploidy accumulating in these lineages. We also see more rounds of polyploidy when polyploid net diversification rate is less than diploids, right? So down here in the bottom, we only ever get 1 or 2 rounds of polyploidy when polyploid species have really high net diversification rates. But up here in the top right, we get 3 to 4 rounds of polyploidy in those higher, in this cases where polyploidy has a much lower net diversification rate than their diploid relatives. That may seem counterintuitive, but what I think is happening, right? If you think about it, if you diploidize fast, and if you polyploidize, and then you diploidize fast, and you get back to this diploid speciation and extinction rate that's high, you essentially preserve that ancient polyploid in the lineage. If you go up and stay as a polyploid, you only ever accumulate 1 or 2 rounds of polyploidy, maybe, if you do really well compared to the diploids, and without any diploidization. I mean, and if polyploids have a much higher net diversification rate, but they have high rates of diploidization, they're going back to a state that has a lower net diversification rate, and they go extinct, right? And so this is the kind of dynamic that we're seeing here, and this is actually very similar to what we observe in flowering plants. So if we look at this top right corner, we see that many of these simulations at the highest, where diploids have a really high net diversification rate, and we have high diploidization, we see that there are 3 to 4 median whole genome duplications in our simulations, even these small-sized simulations, and that's very similar to what we saw in flowering plants. What about the inverse, right? So what about the situations that give us fewer ancient whole genome duplications? Well, those parameter states are where we have lower rates of diploidization and where polyploid net diversification is greater than or equal to diploids. And this is actually kind of similar to what we see in fishes. We know that from empirical analyses, there's not a huge difference in the net diversification rate among polyploid and diploid fishes. They have slow rates of diploidization. And that puts you down into this kind of corner where you get maybe 1 to 2 rounds of polyploidy. And that's the kind of parameter space, empirically, when we go outside, we see that in those fishes. So, so pretty interesting. So I these exploration sims suggest that maybe this dynamic works. But we can go out and look at empirical data and ask how these patterns compare. If we look at, for example, the Brassicaceae, where we sampled more than 50% of contemporary species and estimated net diversification rates and polyploidization rates and everything else for the family, we can plug those into this set of simulations. And so we take our diversification and polyploidization rates from Roman Palacios et al. 2020, and then we use a range of diploidization rates because, again, we don't have an empirical estimate of diploidization. Everything from a very low 0.01 up to 0.9. And we did this with 4,000 taxa with 1,000 replications, 4,000 taxa being approximately the size of the family, and then replicated each of these parameter states 1,000 times to generate our distributions. And we get something that looks like this. So it's a slightly different version of the plot, but on the y-axis here, we have the the x-axis, excuse me, we have the diploidization rate across these different values, and then the mean number of whole genome duplications. And what we see is that there's a slight increase there, but if we draw a line through this, that roughly we see that there's an empirical estimate of about 1.8 average rounds of whole genome duplication within the Brassicaceae, and this is the number of polyploid events. And if you just do tip to root within the family, this is the number of ancient whole genome duplication events on average that we see within the Brassicaceae from our previous analyses. So nice and consistent. And putting this in a different kind of box here, we see that at these higher rates of diploidization is where we're getting back to this empirical estimate of 1.8 rounds of polyploidy within the Brassicaceae, if we summarize across all of those boxes on the previous slide. So again, what we're seeing here is that with those empirical estimates of diversification rate and polyploidization rate, as well as fast rates of diploidization, we're recapitulating our empirically observed numbers of the average rounds of whole genome duplication within these lineages. Okay, well, let's go out and make this a little bit bigger. Let's go and look at our Mayros et al. 2011 paper. This is our science paper on the recently formed polyploid plants diversify at lower rates. Let's take the values from that and look at across an angiosperm average set of parameters for this question. In this case, we used our mean diversification and polyploidization rates that we estimated across those roughly 50 genera in our paper back then. And we used the polyploidization rate of 0.15 from our Wood et al. Estimate back in 2009, as well as 0.3, which we estimated in Mayros et al. 2011 and Barker et al. 2016 using different data sets. And for good measure, let's do a polyploidization rate of 0.45 as well. So this is the rate at which diploids become polyploids. So across 3 different parameter values, 2 of those from empirical analyses. And let's also look at, again, because we don't have an estimate of diploidization rate, an empirical estimate of diploidization rate, let's just use an arbitrary range. So we use this 0.01 up to 0.9 rates. So again, from slow to fast rates of diploidization. And this is computationally kind of an intense thing. We did a lot of work to try to speed this up, but we're going to just simulate to 100,000 taxa with 100 replications each year to look at these dynamics across all these different parameter space, all across this whole parameter space. And what do we see? Well, we get a plot that looks like this. So again, on the x-axis, here's the diploidization rates. And again, the y-axis is the number of whole genome duplications that accumulate from root to tip. And this is just, in this particular plot, just as an example, showing you with that median, that middle estimate of the polyploidization rate of 0.3. And you'll notice that we've got little bars there from 3 to 4. This is empirically what we observed in the 1KP, for example, as well as in this more recent McKibben et al. Analysis, the number of rounds of ancient polyploidy across flowering plants. And what we see is that our, again, here's our 3 to 4 rounds of polyploidy from our 1KP analyses. And what we see is that our simulations are right in the middle of that, especially at the higher rates of diploidization, right? So when we simulate with those empirically estimated diversification rates and polyploidization rates, and when we use higher rates of diploidization, we see that our estimates of the number of, or simulated number of whole genome duplications matches what we observe empirically. And to plot it in a slightly different way to condense that map down into just an average set of heat maps, these mean number of whole genome duplications across all those different parameters. In particular, if we have this rate of polyploid speciation of about 0.3 and high diploidization rates, we get values of number of whole genome duplication events that are consistent with what was observed in the phylogeny. in our genomic analyses, which I think is really cool. We can connect these phylogenetic simulations with these genomic inferences of ancient polyploidy. So what kind of insights, right, did these simulations give us? Which is because these are a tool to explore what's happening, not so much necessarily tell us exactly what happened, but to give us some insights into the biology of what's happening here. And these simulations tell us that all else being equal, high diplotization rates contribute to more rounds of whole genome duplication. And that more rounds, and under these conditions as well, more rounds of whole genome duplication we see more rounds of whole genome duplication when polyploid species have lower net diversification rates than their diploid relatives. And that, I think, importantly, these simulations, though, using these contemporary net diversification rates that we've always held up to be at odds with our observations of the number of ancient polyploidies, these simulations using these contemporary net diversification rates produce numbers of whole genome duplications consistent with our empirical observations across the phylogeny. And that I think that, a big part of this that's missing from our thinking is that diplotization itself is potentially a significant underappreciated driver of those ancient whole genome duplications that we observe across the phylogeny. And so this ability to diplotize fast, get back to a state that has a higher net diversification rate, but preserve that ancient whole genome duplication is important, is an important part of this process. So just sort of restating this question again, if polyploidy diversification is generally low, then how can there be so many ancient whole genome duplications? And it may just be that there's a really fast diplotization, that some of these polyploid events are preserved because they got back to they diplotized quickly and then went on to do great things as paleopolyploids. So what's next for this sort of little line of research here? Well, one of the things we want to do is track multiple states of polyploidy, so these transitions are also captured. But we've got work doing this, but I don't know that it's going to change hugely the results to include things like hexaploids or octoploids. There aren't very many of those in the history of flowering plants, actually. And a lot of the biology does seem to be captured with just diploid and polyploid as the two states. But also, what exactly is diplotization, right? In this model, it's when species return to the diploid diversification rate. But the real world, it's likely a less clear transition. We think about diplotization in many different ways. We wrote an annual review on this a couple of years ago, and you can kind of break it into sort of 3 different large components of when species return to diploid genetics, when species cytologically diplotize, right? When species lose lots of genes. What is the critical biology there, though, that returns species from having polyploid diversification rates to diploid diversification rates? It's not. I don't think it's entirely clear what part of that is important, and then how we measure what that rate is empirically is a open question, depending on what we mean by it. But in the model, it's very clear. So where do we go next? One of the things we're working on in the lab, obviously, is trying to nail down these variation and diplotization processes and outcomes. Work led in the lab now by Michael McKibben and Songyan Kang, looking across patterns of gene loss, looking across patterns of chromosomal evolution, and trying to get a better way to quantify those changes. And also looking across lineages, for example, looking at patterns of chromosome number evolution across the whole entire phylogeny of eukaryotes and modeling how these numbers change. One of the fun things here is led by Jeff Finch, who's looking at evolutionary drivers and consequences of chromosome number evolution and little local species here that we sequenced the genome of from my neighbor's front yard here, Xanthissima grassily. It's we just happen to live in an area where one of the N equals 2 plants lives. And so there's some really fun work there looking at what happens in these lineages, and without getting into too much detail, these are the 1 chromosome, 2 second chromosome of Xanthissima grassily in this nice nearly chromosome level assembly that Jeff has generated, and this is the syntenic comparison to Lactuca. So some really cool work there to come soon, so stay tuned for Jeff's work on looking at diplotization and fractionation in these really small chromosome numbered species of the composite. But I also think the other sort of thing to emphasize here is that the important biology in these in flowering plants and plants in general isn't confined to the period when species are polyploid in quotes, right? When they're just polyploids, because everybody's a paleopolyploid. And so we know, for example, that legacy of paleopolyploidy has contributed to the genetic diversity and domestication and novelty, for example, across lineages. For example, in this case, if we look at the Brassicaceae mesohexaploidy that happened about 15 million years ago or so in the history of the Brassicaceae, it's left a legacy of paralogs and duplicated regions in the brassicas. In this case, here's the genome of Brassica rapa, and you can see all these duplicated homologous regions. And in the past, we've studied this, and I'm not going to get into too much detail, but just to highlight what we see, if we look across, for example, all the Brassica rapa genetic diversity in these lineages, and we identify candidate genes associated with domestication, and then also categorize those genes as whether they're from ancient polyploidy or not. What we find is that these candidate domestication genes in Brassica rapa are enriched with paleologs. So when people went out and selected for traits, without any knowledge, obviously, without any knowledge of the genome, but when they went out and selected for traits associated with domestication, or when they domesticated these plants, I should say, the genes that got pulled along in that process were highly enriched for paleologs. Whether you look at the different kinds of tests, the Swede test, McDonald-Kreitman, differentially expressed genes, or from the literature survey, and when we look at they're all enriched with these ancient paleologs. And when we look at the diversity of those genes, we see that depending on how we identify the paleologs, the paleologs always have, in Brassica rapa, have more genetic diversity than their non-paleolog copies in that same genome. And so if you were to look at the Brassica rapa genome, for example, and ask where's all the genetic diversity, it's in these genes that were duplicated and retained from this ancient polyploidy that happened 15 million years ago. And that makes sense. So that's the case. Then the people, when they selected for traits, they got all this additive variation that they likely pulled in, and it happened to be in these regions of the genome. We see higher diversity to both synonymous and non-synonymous sites, consistent with relaxed selection over time, probably playing a role in these epistatically interacting loci at this point in those genomes, as these genes are maintained in a higher copy number from that ancient polyploidy. But then that gives you this bounty of variation, potentially, or it looks like it does, and that potentially is important then during domestication. And very briefly, just touch on one little study here. How important then is this? How broad is this pattern, right? How important are paleologs for domestication in general? And we have a lot of data for the domesticated species. I think this all applies to species that are not domesticated as well, to natural adaptation, but we have a lot of genomes and a lot of information about these domesticated species. And this is work that Michael McKibben has been doing in the lab, and we'll have this manuscript out hopefully in the next couple of weeks to look at paleologs across domesticated plants. And to do this, I'm not going to get into too much of the detail. He developed a nice machine learning approach to identify paleologs and classify retention status. This came out earlier this year, called Frackify. And this uses machine learning across a whole bunch of plant genomes to extract features and train models to be able to identify these paleologs very quickly. So instead of spending months and months, 6 to 12. Oh yeah. Sorry. Oh, so instead of spending 6 to 12 months to do the likelihood modeling, we can do this in like about 30 seconds. But without getting into too much detail, Michael's work finds that across 22 different domesticated plant species, across a variety of agronomic traits, what we see is that the same pattern we saw in our Brassica work, that these paleologs are enriched, or candidate genes are enriched with paleologs in many, many crop species, and that they're important across this broad range of crops. And so this is a general pattern that we see, that a domestication of crop species has preferentially selected these ancient polyploid derived paralogs in plant genomes, but not other kinds of paralogs. This is not something we see from with other sources of paralog variation. And so it does seem that this is consistent with our analyses of enrichment in Brassica rapa, and that even after millions of years, polyploidy is still influencing genetic variation among contemporary plant species. And so if we think about all those ancient polyploid events that are diplodized paleopolyploids, the legacy of those events on contemporary plant evolution appears to be quite significant. And I really look forward to being able to look at this across many non-domesticated species here in the future once we have more and more data. But just to leave you with a couple of take-home messages today. One, there's a lot of paleopolyploidy in the history of land plants. Maybe if you just do some back-of-the-envelope math, about 8,000 or so more whole genome duplications to be discovered across that phylogeny, given our current discovery rate. Rapid diplotization combined with low average polyploid net diversification explains or appears to explain multiple rounds of whole genome duplication across the history of flowering plants. And that remnants of these ancient whole genome duplications have been really important for domestication and plant evolution and adaptation among contemporary plant species. These regions harbor elevated genetic diversity. And when we look at candidate genes associated with crop domestication, they're enriched for these anciently derived paralogs from ancient polyploidy. All right, with that, I'd like to thank everyone in the lab and funding agencies, and thank you folks for inviting me today. And I'll just leave you with a picture of one of my favorite places out here in Southern Arizona, the Chiricahuas. And I'm happy to take any questions.
Oh, thanks very much, Mike. That was really very cool. I have to be honest, I haven't really thought much about diploidization, but now I'm going to start thinking about it a lot more.
Excellent.
So I did see 2 questions come up in the chat. So I don't know if Kenneth wants to ask them, or if I should ask them for him, or if I can ask some questions. Kenneth?
Hi, Kelsey. Can you hear me?
Yeah.
Can I go ahead?
Please.
Thanks very much. Yeah, thank you. That was a fantastic talk. Really enjoyed it. So I've been wrestling with this concept of diploidization for a very long time, because I'm working on a group where I think it might be playing possibly a role. But just as you mentioned, we've got no idea what the sort of the fraction of diploidization is.
Yeah.
So yeah, I have 2 questions, one directly related to that, but maybe you should ask the first one. You did lay out, so you did a fair number of HISI analyses, and you showed a fairly strong role of a hidden character.
Yep.
But in terms of your BISI analyses, you used diversity, so you didn't actually account for a potential hidden character. Do you think that might be influencing your results in some way?
Well, no. I mean, in those BISI simulations, those were, all just to simulate large, collections of trees, and so there's no real plants there, right? That's not on any empirical data. So, we're using that as a way to simulate those data. And we're getting, sort of distributions when you look at those, from those simulated populations of trees that, they look like the kinds of things you get in the BISI analyses. And so, well, and also things that don't look like that, because we're really pushing the parameter space broadly in those simulations. So I think that empirically, it may be that, polyploid species that do well have these different hidden traits, but they're sort of buried then in that distribution of things, right? And so I don't think in terms of the large scale sort of phylogenetic dynamics, it makes a difference for those simulations.
Thank you. And then secondly, you mentioned that you think that high rates of diploidization have played a role in, say, more ancient WGDs. But do you think it's playing a role in very, very young groups? So say, for example, a group in which there's large amounts of independent polyploidization, so a high rate of polyploidization.
Yeah.
But do you think that it might also be happening, say, in much, much younger groups as well?
Oh, yeah, absolutely. Yeah, I think it's a whole continuum. I think I don't sort of think about a difference there. Absolutely. So those young ones that go on to become old ones, I think that they've got to diploidize pretty quickly. Whatever that means in evolutionary terms, I don't know because we don't have a sense of what that actually looks like. But I think that it's clear from, a lot of our analyses over the years that if you stick around, that we just don't have old polyploids, right? So we have these ancient polyploid events that have been captured essentially in plant genomes, but we don't have polyploid plants that are like 20 million years old, right? That are still not genetically polyploid. And so I think there's, that these things have to go back, even among these lineages where there's lots of recent polyploidy. I think that and no one's done this analysis, but I think those genera probably may produce more ancient polyploidy, right? You have a better chance of one of those surviving, just all else being equal. But that they've got to diplodize quickly, I think, in order to become preserved in the history of flowering plants. So I do think that's really important, and I think that, you know, if you look across some of these lineages like Brassicaceae, where, you know, Martin Lysick's work has shown that there's a bunch of, you know, every time they get into the chromosome painting, right, you see that there's a polyploid in this N=7 lineage or whatever, right? And that, you know, those things are diplodizing fast and then getting preserved. So I think it's really critical to that whole dynamic. Yeah.
Thank you very much. Food for thought.
Thank you. All right. Oh yeah. Sorry. I'll let you handle the questions, Kelsey.
Oh, no problem. Nicola, I see your hand.
Yeah, thanks. And thanks, Mike, for a really interesting talk. So I think it's kind of changed the way I think about polyploidy, you know, because with the diplodization, it's kind of like I guess you could almost paint the branches of a phylogenetic tree, and maybe every lineage, almost every lineage, will have had phases of being in a polyploid state because of the fast rediplodization.
That's right.
So,
yeah.
Yeah. So what I wondered, I mean, I think it looks like every single flowering plant has had a polyploid phase in its history, if you go far enough back. Or am I? Is that
No, that's
right?
right. Let me show you a result that I just got yesterday. Let me change my share real quick. I opened this file up just to do this. It's a little bit easier to see from a recent. This is from a new big analysis that a student in my lab just produced, and I'll share that screen real quick. That puts that into a histogram, which is, I think, a little bit easier to see than the kinds of plots. And so there you go. So yeah, there are some species that just have one, but most things are in that 3 to 4 range. Again, this is a different analysis than the 1KP that I showed. This is a new big analysis using a variety of tools. And so you can see that distribution. There are some flowering plants that apparently have 8 rounds of ancient polyploidy, I guess. So, but most things are in that 3 to 4 range. Yeah.
Right. Okay. So I wondered if, you know, if some lineages are maybe more prone to becoming polyploids, then you could have this, I guess, constant cycling. Yeah. And I don't know if there's any mechanism that would stop that, and whether that would also make it harder to pick up.
Oh, absolutely.
Yeah.
Yeah, absolutely. One of the things that allows us to see some of these events in time in these lineages that have lots and lots of rounds of polyploidy. So, for example, in the Brassicaceae, if you look at Arabidopsis, you can really only see empirically in Arabidopsis itself just those first couple of rounds of polyploidy. After that, it's fractionated to it's impossible to really see it in the synteny or in the phylogenetic data, right? When you look at the gene family structure. But we have lineages that don't have that many rounds that allow us to then sort of peer back further in the phylogeny, right? And lineages that have, and because of molecular rate heterogeneity and gene birth death rates, we have some lineages that are really slow. And so it lets us sort of peer back in time deeper than some of these lineages that are moving really fast. We do know from other work too that we published in 2018 from Landis et al. That those, if we look at the numbers of rounds of polyploidies in the lineages, it is associated with higher net diversification rates. So those lineages that have 5, 6, 7, 8 rounds of polyploidy do appear to have, on average, higher net diversification rates than the lineages with 1 or 2. I think that kind of makes sense, but I think it all gets kind of tied up with rates of just life history and everything else, right? When you look at these, what these species are, these are all a bunch of little annual herbs versus that are really high rates of polyploidy in those lineages versus woodier things down here on the left side. So,
yeah, interesting. Yeah, I guess it almost sounds like it functions as a kind of a speciation pump.
Yeah,
for genome rearrangement and evolution.
Absolutely, and I at this point, and I thought of this way for a long time. It's a big mutation, right? And so, just like any other kind of mutation. Many of them are neutral, some of them are beneficial, and some of them are deleterious, right? And so, and I think of polyploidy in that way, except it's an interesting one in that when you do have these events and then they get preserved, it seems like the net effect, and I didn't show this result, but we do see this correlation of diversity in plant species with the numbers of rounds of polyploidy that they have, purely because of that process where we get extra variation from having all these extra copies, right? And so it may be a big part of the story of why plants are so diverse and so adaptable. And if you think about the experiments and folks have been able to, redomesticate lineages really quickly, or make wind-pollinated brassica rapa in just like 5 or 6 generations, I guarantee if we sequence those genomes and look at, and it's very hard to do, but look at what variation was important for that really rapid, change in breeding systems or mating systems, I bet it's variation from these ancient polyploidies. I guarantee. That would be my bet.
Okay, thanks very much.
Yep, you're welcome.
Thanks, Nicola. And then Mutama, do you want to read out your question, or do you need me to do it for you if it's in the chat?
Thanks, Chelsea, and thanks, Mike. I'm just curious, I fancy to think about domesticated plants and where they occur. And that's just ask the question, if polyploidy is, and I believe that is important for domestication, how does one explain the variation in domestication of plants around that they are not evenly distributed? Is it our ability to discover them, or is there a hidden pattern, either geographical, ecological, or phylogenetic in the pattern? Thanks.
That's a good question. You mean evenly distributed across the planet, right?
Yes.
Yeah. I assume that has much more to do with, with a combination of human culture and history than the plants themselves. And the plants that we continue to use today and how those spread in time, more than anything about the distribution of polyploidy itself. Phylogenetically, we have, domesticated plants are not randomly distributed, but concentrated in a handful of families, really, right? And I think that probably has more to do with the traits of those families themselves than necessarily the polyploid history. But they are all notably not in the distant past polyploids, as most things are. But Itai Mayros and colleagues a few years ago showed that phylogenetically speaking, that polyploidy was a significant precursor to domestication, right? And I think that our results on this legacy of paleolog variation from those polyploid events, that increased variation is potentially what explains that phylogenetic pattern that Itai and colleagues demonstrated back in 2016. So that's, hopefully that answers your question. But I think probably the distribution across the planet has much more to do with the human history and our contemporary uses of these plants more than anything else.
Thanks, Mike. It does it. It still leaves me the question of why not Southern Africa? Thank you.
Well, yeah, absolutely. I mean, I think that if you look at a lot of these regions, though, too, and you get into, this is not necessarily my area, but there are, all these fascinating plants that people no longer use. So, for example, here in Arizona, there was just a paper that came out a few weeks ago on ancient, these sort of domesticated agaves that we no longer use, but that indigenous people had domesticated and planted. And actually, if you go out in some of these sky islands here, or mountains here in southern Arizona, you'll see these sort of as a botanist, you notice these patterns. They'll be like, oh, here's a bunch of agaves, and I don't know what these are. It turns out that there's strong evidence that those are from ancient indigenous, plantings from 8,000, 10,000 years ago. And that they were abandoned at one time when there was a major drought or something, and there's turnover in human civilization here, and that these were historically domesticated things. So I think that like I said, our contemporary domesticated species are a fraction of what humans have done, right? And so over time, and so there very well could be some interesting patterns in these abandoned or these sort of lost or forgotten domesticates. So yeah.
Thanks, Mike. If anyone has any other questions, please either type them in the chat or ask them quickly now. I am being conscious of time, so.
Okay. Well, thank you so much. That was wonderful.
Yeah, thank you very much, Mike. It was really great to think more about diploidization as opposed to polyploidization. So our next speaker is Ruan, and he is going to be talking with us about some of his work in Cyperaceae. Thanks, Ruan.
Thanks, Kelsey. Well, I suppose first thing first, we need to share the screen. It's always that awkward little pause while someone's looking down at their screen while they're doing it. Cool. Can you guys see my screen? Can you also see Zoom things on top of everything?
No, it all looks good.
Oh, brilliant. Okay, that's great. Hi everyone. Thank you, Kelsey, for passing the mic to me. My name, as has been said, is Ruan. I am though technically I haven't had the hat put on my head yet, I am a recently finished my master's at UCT, where I worked on the ecophysiological and ecological consequences of, to a lesser extent, ploidy variation, to a greater extent, genome size variation in two groups of Cyperaceae that are very species diverse in the Cape, down here in the Cape floristic region. And so I'm preaching to the choir here. This has already is a given, given the topic of today's talks. But as we know, genome size is incredibly variable within the plant kingdom. Relative to eukaryotes in general, is some of the largest sort of range in genome size available among eukaryotes. So this is deliberately on a linear scale to emphasize that variability. Our genomes are a measly 2.9, say, let's call it 3 gigabase pairs, which just shows you how big Paris japonica's genome really is. And much of that variation is obviously attributable to small, incremental, stochastic changes in the length of the genome, but also, the topic of today, large-scale changes, chromosomal duplication, loss of chromosomes, and whole genome duplication, which is where polyploidization comes in. And so, ordinarily, even among polyploid biology, we are interested in the functional consequences of the genome. We are interested in what the instruction manual of organisms, cells, tell the cell to do. The length, however, of the instruction manual is also important. This is sometimes called the nucleotype properties of, as opposed to the genotype and the phenotype, properties of the genome that are attributable to its length, its arrangement, its karyotype. And the analogy I use is that, like an instruction manual, it can be a pamphlet or a tome. You can have a collection of copies and duplicates of instruction manuals, and if you're not careful and you hoard too many of them, as the plant kingdom has done over evolutionary time, you end up with a whole wing of your house dedicated to housing instruction manuals. And so, in this metaphor, the nucleus and the genetic material therein, it takes up physical space. Cells cannot be smaller than their nuclei. Nuclei cannot be smaller than however big they need to be to contain all that chromatin fiber. And an organ in which this is particularly obvious to measure and easy, especially for undergraduates to measure with the microscope slide and nail polish, is the stomata. Another benefit of the fact that this is so easy to measure in stomata is also that stomata are really, really important in plants because they are the gateway through which all water relations between plants and the atmosphere happen. The ability of stomata to open, close, how far apart they are from each other, how big the stomatal aperture is all affected by how big how big the guard cells are, the rate and opening of closure as well. And now, because genome size affects cell size through the nucleus, cell size affects stomatal parameters in such the way that larger cells mean that your stomatal pores might be further apart. Smaller cells will mean they're closer together. Larger guard cells, larger stomatal aperture, etc., etc. Now, down the chain of biological organization, or up the the ladder of hierarchy, depending on how you want to put it, this can have consequences for the ecology and the physiological function, at the very least, of plants. Where I investigate aspects of this, I say aspects, not everything, hence the sum of in the title of this presentation, is in the shenoid sedges of the Cape Floristic region. So particularly the feinbos species. These two genera, Schinus and Tetraria, they're not each other's sister genera, but they are the two most species rich in the region of the tribe from Cyperaceae. Some cursory notes: Schinus, generally smaller, unnoded culms, basal leaves, sometimes reduced, and a terminal set of inflorescences, whereas Tetraria are anything from this big to 2 meters tall, noded culms, big, quite robust leaves in some species, and a paniculate set of inflorescences at the end of their culms. Among the Tree of Life, drawing on a paper by Elliott and colleagues, here we have the cyperid clade, so that's Cyperaceae and its sister sort of species, poor relatively, families Juncaceae and Terniaceae. And plotted on this are the bars represent genome size. And so pretty quickly, it becomes quite apparent there's an awful lot of not much happening in the two larger tribes of Cyperaceae, and indeed in most of Terniaceae and Juncaceae. But there's an awful lot of a lot happening in Eleocaridae and Schini, which is primarily in their namesakes genera Eleocaris and Schinus, where genomes are larger and more variably so. Zooming in on Schini, the tribe that I work in, the two Cape lineages pointed out here by asterisks, you can see Tetraria, teeny weeny genomes, not very big at all. Schinus, enormous, variable, messy, spoiler. Now, Schinus does have species that are not present in the Cape. It's quite a big genus, actually. But the Cape species do represent a clade. And so they can be compared as such. Tetraria has a few species not depicted here that occur outside of the Cape, sort of, and these two groups here of more or less equal size, despite the vertical scale on these two phylogenies, are what I compare. Because in the Cape, things grow in a common environment, at broad scale at the very least, and so that warrants their comparison here as well. Sort of the ghost of this underlying mechanism is haunting our expectations here, in that we think about how if genome size is big, so would stomata be big, so would stomata be further apart, so would the conductance, glossing over quite a bit of ecophysiological sort of assumptions and reasoning there. Stomatal conductance for a given unit area and all else being equal would be lower. We might then also expect that their ecophysiology is more conservative, more slow, use what you have, but don't assume you're going to get more. Whereas Tetraria, with the inverse of all of these features, might be more acquisitive, with higher level of conductance, more focused on nutrient acquisition. If it's there, take as much as you can. There will probably be more, is the expectation. And then additionally, this comes into the question of whether these consequences are manifest, is that Schinus have, shall we say, a more culmy body plan than Tetraria. The culm is greener, it's unnoded. We expect that photosynthesis and ecophysiological strategies will be more apparent in the culms. So while I said they're not each other's sister lineages, we do still need to compare them within a common framework. And because that's quite difficult if you stitch different phylogeny reconstructions together that are based on different genetic markers, we reconstructed one for the Cape members of the tribe in a common ultrametric beast reconstruction. And then we could do phylogenetic comparative methods with it. And so that includes an existing data set, which we supplemented with flow cytometry data for genome size estimates, and trait data based on both newly collected and existing specimens. And here you can see scanning electron microscopy. I can recommend it. It's great for presentations because you get pictures. So genome size, pretty obviously, the first key thing we need to double check. It is already known to a certain extent, but it's nice to just see it visualized. Tetraria have small genomes, genus have big genomes. But within each genus, there are also patterns. I won't go into those details too much, but those patterns are consistent with the fact that we know the genus is full of polyploids, where sister lineages are expected to be more different in genome size than you would expect by chance. Does this reflect in guard cells? Ding, 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 yes, it does. The culms and the leaf tissue of both well, where genus has leaves, some species don't. Those are obviously not on this slide. Species with big genomes have bigger guard cells and stomata that are further apart. Species with small genomes have closer together, smaller stomata. Awesome. In a more analytical framework, we also have a very assumption-heavy, but still fairly, we can interrogate those assumptions. We don't have time to do that here. Turning guard cell length information and density information into an estimate of maximal conductance to water vapor. That's also lower in species with larger genomes, both, and this is significant in a PGLS context as well. So the interrelatedness based on the phylogeny does not mop away the pattern. And so we can put lovely, satisfying green ticks next to most of these patterns that we would expect. The slightly more complex ones, though, require some additional data. We collected the ecophysiologist and ecologist's best friend and worst enemy, specific leaf area, along with something close to an analog of it called specific culm area, which we did for all species, based on the fact that some species don't have leaves, so we had to do something for them. And then we had carbon-nitrogen ratio data for leaf and culm tissue as well. And for these metrics, higher SLA is more acquisitive. Higher carbon-nitrogen ratio means more structural, more cellulose per unit nitrogen, as opposed to more photosynthetic, greener when you've got low carbon-nitrogen ratios. And what we find is that genus species with their big genomes tend to have more photosynthetic tissue in their culms, or rather, their culms are more photosynthetically similar to their leaves, whereas in Tetraria, the triangles on the left of the bottom plot, their culms are structural and their leaves are green. That's the interpretation that we've gone with. I say gone with. It's what I interpret from these data. Secondarily, there is a small discrepancy between in general, the specific leaf area of leaves of genus and Tetraria, and this is significant, in that on average, the specific leaf area of Tetraria leaves is a little higher than the specific leaf area of the leaves of genus. So never minding the culms, the Tetraria leaves are more acquisitive on average than any tissue in genus. And so we have weak and caveat-rich evidence for these expectations, but it's difficult to disentangle these things from the aut ecology of these groups. We have one polyploid-rich group, no polyploids in the other group in Tetraria. And so we don't have an awful lot of power here to separate in this study these effects from the fact that these two genera are phylogenetically distinct. This does then limit the ability we can make inferences about what this means in general, but it's still nice to think about it and still think about genus and Tetraria in the context of broader patterns regarding traits and biogeography. So we know that in quite a few groups of plants, actually, the polyploid lineages tend to prefer the arid areas of a given biogeographic context than their diploid progenitors. But then in other groups, the reverse is also true, either due to the fact that they are less efficient in using water due to mechanical constraints like the timing it takes stomata to open and close, whether or not they can respond to environmental fluctuations in a timeless fashion, or if they just need more water because of some other advantageous trait that has changed. When polyploidization occurs recently in a lineage, there's an awful lot of the trait spectrum of a plant, I think, that shifts suddenly. As opposed to not even paleo polyploidy, but polyploidy that occurred in less recently, the trait spectra of a species has time to adapt. Or not even adapt, it has time to accommodate this new large genome that lives in every single cell, this mechanical constraint. And then with further evolutionary time, we have rediploidization. And again, this is contingent on taxonomic scale, biogeographic scale, spatial scale as well. So landscape versus across a region. Also, we have the fact that character change and the introduction of evolutionary novelty is often a consequence of polyploidization. Well, there's more DNA in every single cell for natural selection to act upon. Conversely, there is also a smaller trait space in principle, in that cells now cannot be very small if your genome is very big. If your genome is small, your cells, your tissues, your organs, the cells can be as big or as small as they need to be. It's variable, but now there's this lower limit placed on cell size when your genomes are big. And so you can argue, and many authors have, that this places a limit on the available trait space in lineages that have recently polyploidized. And so in a very biology kind of way, which is it, do polyploids like hot, wet, dry, or cold places? It depends. You can read more about this and some of the other data we presented in something that is in review. And as usual, we have the logo slide to thank all the disparate data sets and funding over the years, and to SASSB, who sponsored my attendance to the most recent of our conferences, which is what this essentially was this presentation. And thank you to the organizers of this webinar for inviting me. And then, of course, thank you to all the other odds and sods along the way. And if you have any questions, please do ask. Thank you very much.
Great, thanks, Ruan. It's really, I remember talking to you about Cyprus many years ago, it feels like now, back in like 2019. So it's very cool to see what you've been up to since then. I was wondering about your taxa, and sorry, I'm going to jump in here. If other people have questions, please ask, but sometimes I can't always help myself. So, so one of the things I was wondering with your two different genera, did you have one that was slightly more like polyploid rich, would you say, or did like each were there? I know you focus more on genome size, but were there more polyploids in one genus versus the other, and did you notice any patterns in that context?
So, there's two answers to that question. The one is, I didn't investigate ploidy variation and like ploidy series in these analyses at all. And I would have loved to, so it's not entirely me saying, oh, this is beyond the scope of the study. It's not a complete cop-out. We do not have ploidy information for Tetraria. Tetraria, anecdotally, you can speak to Tammy Elliott, one of the co-authors, and was in our lab, a postdoc. The Tetraria chromosomes are so tiny. So when you do chromosome staining to do counts, she can't reliably tell if they're on top of each other or not. And so it's very difficult to get a count. And that's a huge impediment to thinking about this. A third of the two things I said was an answer to this question is that Cyperaceae are holocentric. They have holocentric chromosomes, so they have distributed centromeres. And that's like a whole avenue of complexity to add to this. The relationship between ploidy level and genome size just goes completely out the window in holocentric organisms. At broad taxonomic scales in non-holocentric organisms, it's a diffuse relationship because you have rediploidization happening over evolutionary time. in holocentric organism, that relationship goes out the window, and in a million years it's gone. Who knows? You can just have all your chromosomes split in half and it's all perfectly valid. Nothing goes wrong. Quite strange.
Oh, that's cool. Thanks. We have time for another question or two. I see Kenneth's hand. Go ahead, Kenneth.
Thanks, Kelsey. Thanks, Ruan. That was a really interesting talk. I'm interested in, so one of the things that is sort of used as a potential explainer for limiting large genome size is specifically a lack of phosphorus. And the Cape is notoriously poor in phosphorus. Did you consider, say, phosphorus concentrations in the habitats of these environments? Did you measure phosphorus amounts in the sedges? Any idea what might be happening there?
So that was beyond the scope of my study, actually. No, so we didn't measure any phosphorus. You are quite right. That is one of the many ideas floating around about what can limit this sort of thing. It has been explored by Tammy and Muthama and Peter Burish, but at broader scales. So not necessarily at the individual plant level, more at the scale of the known habitat of an organism. You have, soils grids, you have regional scale soils data. We don't have as much direct phosphorus data for this. And that's a limitation, because phosphorus at the plant tissue level is, especially when you have microhabitats involved, I think can be quite variable. And it needs to be paired with nitrogen for it to make sense in the context of nutrient limitation on genome size in general. And we don't have that data as much as well either, because then you need to pair it with like the available soil nitrogen in the spot the plant was growing. We just haven't, that data hasn't been collected, unfortunately.
It's a possible PhD opportunity.
Yes, very much so.
Thanks very much.
Thanks, Kenneth. And then I see Nicola's hand, and then after Nicola, we'll move on to Henny.
Yeah, thanks. Thanks, Ruan, for another great talk. I thought you raised such an interesting question, actually, and maybe Mike has some insights here, because you're talking about sort of rates of trait evolution linked to, I guess, sudden polyploidization, and then Mike spoke about diploidization being very rapid. So I wonder how the rate of diploidization, how fast that rate is relative to rates of trait change, and if anyone has any insight into that.
Well, I'd open the floor for Mike there.
It's a good question. I'd say that I'm just thinking off the top of my head to studies where we've looked at, estimated trait change from phylogenies, right? It's probably on a similar order, but I think the trait change, is probably, that's a good question. I think the rates of diploidization are wide enough as well as traits. I mean, they're probably on a similar order of scale, though, I would say. And some of these traits, we've looked at ecological niche differentiation, for example, in polyploids versus diploids. If you think of that as traits, right, as an ensemble of traits, it can happen really quickly, right? With this sort of biology that Ron was talking about there, with you get these big changes in cell sizes and all that cytonuclear effect that just happens, and you can get these big changes in the water relations and other stuff that you did a wonderful job discussing. And so that could be like almost sort of in a phylogeny instantaneous, right, if you think of it like that, and actually in a greenhouse. So the traits can change really quickly in comparison to how fast that diploidization process probably happens. But a lot of other traits, if you think about things that are adapting, it's a good question. I don't know that we have a great empirical estimate of those two things, but I would say ballpark probably similar, at some scale. Yeah, I don't know.
Yeah, no, fascinating.
Take my lack of assertion, my lack of confidence to reflect the lack of confidence in that. How's that? Yeah. really wonderful talk.
Yeah, thanks, Ruan. Thanks, Nicola. And then our last speaker for this webinar is Henny Niemann. And so Henny is a PhD student in my lab. So Henny is going to tell you what we've been up to lately. Thanks, Henny.
Okay, thanks, Gabi. Let me get this the screen shared. Okay, let's go there. Hold up and get that going. Perfect. Can you guys see it?
Yep, looks good.
Okay, fantastic. Thank you very much, everyone, for the opportunity to talk to you guys a little bit about this passion project, which was a bit of a distraction actually from my PhD, although it was quite important for informing some of the work that we were doing, that we have been doing for my PhD. So my talk today is going to be about this paper we recently published, the title of which you can see on the screen. However, since our theme today is about polyploidy, I'm going to have a little bit of a side quest within this paper, one genus or two insights into the impact of cryptic polyploidy on systematics with the Hypoxis parvula species complex as a case study. So, firstly, I want to introduce you guys to our little research group. We are the Polyploid Evolution and Speciation Lab, and we are quite interested in studying cytogeography, flower color polymorphism, polyploidization mechanisms, and hybrid speciation, particularly through the lens of our favorite genus, Rhodohypoxis. We really like this genus for a couple of reasons, mostly because there is polyploidy present. It's quite easy to maintain, and it's quite a tractable genus. With 6 species and with about only 2 species containing most of the polyploids that we are interested in. So I mentioned that it's a tractable genus, but this might have recently changed. A study in 2011 by Coates et al., based on chloroplast DNA regions, actually found that Rhodohypoxis is embedded within Hypoxis, making it a much trickier and less tractable group, since Hypoxis is basically the largest genus within the Hypoxidaceae. And because of this and hybridization, which has been noted between Hypoxis and Rhodohypoxis, particularly the ones within this phylogeny. In fact, the ones in this phylogeny are from a population with known hybrids. Last year, Rhodohypoxis was actually sunk within Hypoxis, and obviously us PEAS group members are quite upset about this, because we are quite interested in studying this group because of this tractability. And so besides all of the morphological differences between morphological and ecological differences between them, we also figured it's feasible that ploidy changes, which obviously, as I mentioned, Rhodohypoxis is known to have ploidy, could have contributed to the breakdown of reproductive barriers between these two genera at this contact zone where these 2 taxa within this phylogeny came from. Also, when using chloroplast DNA markers, a diploid's first hybrid exclusion approach is recommended, since the chloroplast is maternally inherited. And thirdly, work within our lab actually demonstrated that the Bowery var confecta from Sentinel is a diploid population. But very little is known about the cytology of Hypoxis parvula, which in and of itself is a bit of a species complex which has variously been treated by numerous authors. And this is basically the starting point of this sidetrack paper of ours. So the Hypoxis parvula species complex, which we then studied a bit more in detail, as it was before our paper, consisted of 5 soft-leaved taxa. As you can see, they don't necessarily represent what you'd usually think of when you think of Hypoxis, which most people think of large flowers. These are very small little plants. Hypoxis membranacea and Hypoxis parvula albiflora are commonly confused with one another because white flowers, which is also quite rare in the group. Then Hypoxis parvula parvula and Hypoxis limicola were recently subsumed, not that recently, but were subsumed because of the differences between them being largely flower size. Potential Geiger's effect, anyone with a polyploid knack would immediately think that. And then finally, Hypoxis uniflorata, which basically was just poorly known. The little bit of material we had available in herbaria basically suggested it looks quite a bit like Hypoxis parvula, but there was not enough to go off of. So we started off with basically studying this group. So the major aim of this study was basically to determine the taxonomic status of the lineages within this species complex using flow cytometry, morphological, environmental, and molecular data. But sneakishly, our side quest aim was to determine if diploid's first hybrid exclusion sample approach could improve the resolution of the chloroplast DNA phylogeny of Kosian. So the methodology was first obviously collecting all of our plants from this Parvula complex, as well as a bit of rhodohypoxis, specifically diploid rhodohypoxis. So we collected a minimum of 10 plants per population from 14 different populations across South Africa. As you can see there is our taxa with the amounts of populations we sampled material from. Then we went on to do some flow cytometry. We used some DAPI stain nuclei according to the pseudo-Tavernic approach to be able to calculate some DNA indices and maybe get some idea of what's going on in the genomes of these taxa. And then we also went on to extract DNA only from taxa that had the lowest relative genome sizes based on this flow cytometry data. And then we produced a new phylogeny, a maximum possibility, maximum likelihood, and Bayesian inference tree phylogenetic analyses were then conducted in order to check out these guys. So firstly, let's get the less interesting stuff out of the way. So Hypoxis uniflorata, as expected, was something completely out of the Parvula species complex. It was, like I said, it was only really ever collected once before we went back with a lot of effort to try and track them down again by Justice Thode, and we actually found that it played it closer with other Hypoxis. And this matched with some of the morphological characters we were able to assign to it, including having a pyramidal stigma instead of a spherical stigma, which is actually diagnostic of a lot of the taxa within the Parvula complex. Then also, Hypoxis membranacea was a bit interesting. It actually clade far away from the Parvula species complex, and its resemblance to other taxa within the group seems to be a bit of a convergent evolution situation. It actually claded closer with Hypoxis angustifolia, which is another forest-dwelling species. Hypoxis, those of you who know, are generally grassland species, and it's quite interesting that these two taxa, which are among the very few forest-dwelling taxa, claded with one another, and they were also actually reported to hybridize with one another as well. Then onto the Hypoxis parvula and Hypoxis limicola situation. So as I mentioned, the flower size was a big difference between them, which was initially what prompted Hilliard and Burt to separate these two taxa, and obviously that's not necessarily enough. But we found a couple of very interesting additional things as well. So when we looked at the relative genome sizes of these taxa, basically via DNA index, we found that Hypoxis parvula is actually quite a polyploid series in and of itself. Both the varieties, Hypoxis parvula albiflora, which is the white-flowered version, and the yellow-flowered version have at least 3 different DNA indices, obviously indicating a bit of a polyploid complex going on there, which is not unusual for Hypoxis, so this is a little bit to be expected. But Hypoxis limicola, interestingly, didn't actually match any of these, and it basically fell into its own little DNA index, and in fact it doesn't seem to be very keen on crossing out at all. It is very self-compatible, and so long story, I'll get to that now. But this little arrow that I'm pointing at here, this is actually the genome size of the Hypoxis parvula that came from Sentinel. So it is clearly also it's most likely a polyploid, so it's probably good that we resampled and looked at this phylogeny again with a new sample that was actually diploid. So we had to also think about why would Hypoxis limicola's flowers, because clearly they are closely related, why would these flowers be so small? And as I mentioned, they are quite self-compatible, and we are actually thinking it got these smaller flowers because it increased. Smaller flowers have a smaller proximity of the anthers to the stigmas, and a little bit of emasculation studies showed that it is self-compatible. It does need to pollinate itself, but it is fully self-compatible and almost always is found with lots of seeds, something that you would not find in Parvula, which is completely self-incompatible. There are those little seed capsules in the hypoxis limicolar plate. Then lastly, the rhodohypoxis barii. Here we can actually see that our diploids first trying to avoid these hybrids approach clearly made a bit of a difference in the sense that we now have some resolution. Clearly, rhodohypoxis barii is sister to the hypoxis parvula and hypoxis limicola group with quite high support. However, this does not resolve the polyploidy, the polyphyly of hypoxis, but it demonstrates that chloroplast DNA-based phylogenies are significantly impacted by the presence of polyploids and hybrids, as one would predict. With so much polyploidy reported in hypoxis, which is obviously needed now, as we can see, for understanding rhodohypoxis, the polyploid hybrid exclusion sampling approach becomes ultimately impractical. So what conclusions can we make from this little study on like the parvula complex? So the first thing is that we have now evidence for a new a polyploid series within the hypoxis parvula. And it's quite a nice system to work with because it seems to be quite consistent, and we have a couple of characters that can separate them out. So it might be, potentially replacing our rhodohypoxis love. And collectively, the data from this study indicates that hypoxis limicola and hypoxis uniflorata are indeed distinct taxonomic units and of unique evolutionary lineages. Thirdly, polyploidy hybridization has a significant impact on the chloroplast DNA-based phylogenetics. This is something that we kind of didn't know. We just really hoped that we could get away with an easier approach, but more sophisticated methods such as utilizing low copy number nuclear genes together with cloning or next-generation sequencing are crucial for overcoming these challenges and increasing the robustness of phylogenetic reconstruction in polyploid-rich groups. So coming soon, this is the actual PhD project, not the side project, is actually in the works of producing a rad-seq phylogeny of the Hypoxidaceae of Southern Africa, where we'll hopefully sort out some of these challenges and hopefully have a very cool system to work with in regards to that. And with that, I would like to say thank you for your time, and I would love to take any questions if you have them.
Thanks, Henny. I like that you pitched that as a side project.
It is a big side project, but
I like your distractor.
Yeah.
I see Kenneth's hand. Go ahead, Kenneth.
Thanks, Kelsey. Thanks, Henny. From a just purely ignorant sort of side, are there any nuclear-based phylogenies of the Hypoxidaceae that might sort of support your contention that rhodohypoxis and hypoxis, one doesn't need to be sunk into the other?
Yeah, unfortunately, there are none that are published. We have looked quite a bit, and unfortunately, so just because of all the polyploidy, there could be similar issues with nuclear. As I mentioned, you oftentimes will have to do some cloning if you are doing these polyploid-based phylogenies. So unfortunately, not as far as we're aware, there's no real nuclear data available yet. And because we know how polyploid hypoxis is, it seems a bit smart to maybe, go the other route, unless obviously you have the time to do flow cytometry on every Hypoxidaceae of Southern Africa and start off with that diploids first approach, which sometimes could be a little bit impractical as well, because it would take a decent amount of time.
Thanks.
I'm also hoping. Sorry, go ahead.
Kelsey. Just a quick follow-up question. Your rad-seq sampling, will that be sampling polyploids as well or not?
Yes, it would. We are trying to sample as much as like as broad of a range as we can. We are. It is obviously also better, even in rad-seq phylogenies, to try and keep the polyploids to a minimum initially. But because obviously polyploidy is such an important component of speciation, you can't really do phylogenetics by avoiding polyploids. This is actually also a big problem with these with this approach of the diploids first, because at the end of the day, as we've heard quite a bit today, like polyploidy is such a crucial component to speciation, and it's such an interesting part of phylogenetics. We are actually only really starting to properly delve in now that the techniques are becoming more accessible and available. So I don't think avoiding polyploids is actually ever the answer, because that's where the cool stuff is. So yeah.
Mike, I see your hand.
Yeah, thanks. That was a wonderful talk. I was just thinking, I had a couple of questions real quick. As you sort of look ahead at the RADseq analyses, do you have a genome or a collection of transcriptomes or sort of something that
We
you're going
actually
to
have a
back to?
transcriptome available, thank
Cool.
goodness.
Yeah.
Also, just lucky because in this group there are some medicinal plants, and again within the Southern African context, obviously, and any context anyway, like something that is of medicinal value gets preference when it comes to whole genome. So we were very lucky in that regard, having a transcriptome in order to actually pry this apart, and we are in the process of working with that in order to hopefully get that going.
Fantastic. And do you have a sense of, I mean, I know you're still doing this, but just thinking about the sorts of analysis that you're going to run to try to include all that complexity? Are you looking at, yeah, I was just sort of curious what your first take might be on the RADseq analysis. I don't have it. I think there are several ways that it could go. I'm just sort of curious. Yeah.
Yeah, so the first thing, obviously, we just want something a bit more resolved than the current hypoxis phylogeny. That is the first step. We are quite interested in, again, like using this phylogeny at the end of the day in order to answer a couple of questions in regards to what is the polyploidization mechanisms within South Africa. See, these genera are so interesting to us because of the distribution and the distribution of polyploids among them. As I mentioned, I think I had a quick picture of them, but the Hypoxidaceae of South Africa, there's about 4 major genera currently recognized, and polyploidy isn't perfectly distributed across them. Some genera have massive amounts of polyploidy, and others are very stable. And we're thinking there could be some interesting environmental things to look at in regards to what is driving this immense polyploidy in hypoxis and a little bit less in rhodohypoxis. And then going, obviously, as Kenneth also mentioned earlier, the Cape, where a lot of these, where 2 of the genera are mostly based, is notoriously low in polyploid poor. So we could also maybe use this in order to try and figure out if that same impact is happening within the Hypoxidaceae, or that same thing is happening here. So there are so many questions. The first thing is obviously just to get a resolved phylogeny in order to start to be able to ask these questions and do these types of analyses.
Yeah, that's great. There's a paper that comes to mind from Leonie Moyle's lab looking at some of this in the Solanaceae and in Solanum in particular, where they used, resolved all the sort of species level phylogeny with this kind of not RADseq, but different sorts of nuclear data that I think is, potentially a really cool example of how to kind of get around all the incomplete lineage sorting and everything else that's going to be in that tree when you try to figure out all the relationships. Anyway, I think that it's really cool, but I think this is also there's a lot of fun challenges here. How's that?
Oh, yeah. No, definitely.
But it's going to be fun. This is so cool. Thank you. Yeah. Good luck.
No problems. Thank you, Mike.
Thanks. Mutama, did you want to ask Kenny, or should I answer for him?
Yes, I'll come in. I'm just curious on especially that question of the study limited to Southern Africa, and just thinking of where I've seen hypoxis in the continent.
Yeah,
I'm wondering whether we're not missing opportunity to see bigger pattern and complexity of pattern, especially as you start going across these savanna divides which don't have hypoxis, which is my impression more in the cooler, higher elevations separated by these low areas.
Yeah. Absolutely, that's a very good question. And again, hypoxis actually has 2 centers. Obviously, Southern Africa is actually one of the primary center of diversity and endemism of the Hypoxidaceae. But there is a secondary one in Central Africa. Again, we have to start somewhere. And again, it's a PhD. It would be great to do a much broader sampling, a much broader study. But this is a starting point. This could at least help us to look at it at its core, where at its center of diversity and endemism, and then work our way out. So this is a start. This is only supposed to get us to start looking at some of these patterns. But I absolutely agree. A much broader study that goes far beyond Southern Africa is definitely warranted, and hopefully this would give a bit of a blueprint or a baseline for that sort of study to be able to be done.
Okay, if there are no other questions, I'd just like to thank Mike for taking time in the early morning to be here. Henny and Ruan, thank you so much for sharing your work with us. And I just appreciate everybody spending their Thursday afternoon with us talking about polyploidy. So thanks, everyone, and have a great afternoon. And Mike, have a great rest of your day.
Yes, thank you. This is the highlight of the day. It's all meetings from here on out. So I appreciate it.
Yeah, well, it's a good way to start a day then.
Yeah, really wonderful to hear about all the research that Ruan and Henny are doing. That's great stuff. Thank you.
Thanks, and hopefully we can host you sometime in South Africa.
Yep, I hope so too.
Cool.
Thank you very much for our as well. Yes.
Thanks, everyone.
Thanks, Fanis, for hosting as usual. Much appreciated.
Thanks. Bye.
Thanks, everyone. Bye-bye.